Hello, everyone, and welcome to PPMD's Biomarker Initiative webinar, where today we're going to be talking about leveraging data for new discoveries. I'm Eric Camino. I'm PPMD's Vice President of Research and Clinical Innovation. Joining me on the call today is Pat Furlong, PPMD's founder and president. We're going to be joined by two speakers today. Dr. Glenn Walter, who's a professor of physiology at the University of Florida, who's going to be giving our main presentation today. And as well, we're joined by Dr. Krista Vandenborne, also from the University of Florida, who's going to be available during our question and answer session. So what we're going to be discussing today and kind of our main presentation is some of the work that has been ongoing through the ImagingDMD natural history study, and how we are looking to support the leveraging of 10 years worth of natural history data from both ImagingDMD as well as Leiden University to make new discoveries in Duchenne, specifically working for this Duchenne protein mapping project. Now, before I hand it over for a presentation from Dr. Walter, I wanted to give a little bit of insight into PPMD's biomarker initiative. I'm sure many of you have seen the emails and the blog posts that we have been sharing, but for anyone who maybe isn't as familiar, just to give a quick overview. PPMD's biomarker initiative, if I can, give me 1 second. There we go. Our biomarker initiative is really a kind of a focused research effort to support work that is looking towards the discovery and validation of biomarkers that can enhance the care of individuals with Duchenne and Becker muscular dystrophy, as well as improve drug development and help to accelerate clinical trials so we can get to therapies faster. And as you'll see in the talk today, biomarkers have many different forms and uses. We're going to hear about some imaging biomarkers that can be used to help understand disease progression, talk about how proteins can be used to, monitor for safety or understand, again, help us with disease progression in Duchenne. So these are really powerful tools that we can use to, accelerate our path to therapies, and it's something that we want to support. We have, through the year, supported a number of projects that have worked towards this. And in 2020, we've had a couple of really positive announcements towards the validation of biomarkers or new projects that we're supporting. And I just wanted to highlight a couple of those. So in 2020, over the summer, we had some announcements from the Duchenne Regulatory Science Consortium. And for those who are unfamiliar, this is a consortia of academics and industry members. PPMD helped to co-fund with the Critical Path Institute, that they work together to enhance clinical trials. And so this summer, DRISC announced some of their fantastic progress on developing some safety biomarkers. So they have 2 different safety biomarkers they're working on. This is a drug-induced liver damage safety biomarker, so a really sensitive marker to look and see if an investigational product might be causing liver damage, and a similar safety biomarker for skeletal muscle, although that one's actually about a 4 protein panel. But they are making great progress. You have to go through and work with regulators in the FDA to get to a validated biomarker. And so we're really pleased with the strides that have been made there and look forward to seeing how they continue to move forward in 2021. And then this fall, we announced a project to support a non-invasive urine biomarker for assessing muscle mass in Duchenne patients. And this is actually a collaborative project that is being funded by PPMD in partnership with Duchenne UK and MDA. So we were really pleased that we were able to work together with some of these other foundations in the Duchenne space to drive forward some novel biomarker development. And so again, that'll be something to look out for in 2021 as that project kicks off. And if you want to learn more about any of the biomarker projects that we've been supporting or the biomarker initiative, you can find that information on PPMD's website. Now, before I hand it over to Dr. Walter to give his presentation today, we have a short little video on some of the work that has been done at ImagingDMD for those who are unfamiliar. I know many people on this call have probably participated in the ImagingDMD natural history study, But for those that haven't, this is a nice little vignette that really encompasses the great work that's been ongoing there.
The goal of ImagingDMD is to develop MR imaging biomarkers that allow us to accelerate therapeutic development. We are working hard to develop biomarkers that are very sensitive so that we can have trials that are shorter in duration and can be done in a smaller patient population. Another part that's really important is that we really want to have a definitive readout of a drug. And the MR images provide a picture that is not influenced by motivation. The data are the data, and that is really important when you're doing clinical trials. The imaging biomarkers that have been developed as part of the ImagingDMD project have already been used in a number of clinical trials to help make a go, no-go decision and really provide an unbiased readout of the efficacy of a number of different drugs. As part of our growth, ImagingDMD moved from being a NIH study at 3 sites to now having a clinical trial network that involves 30 sites across the United States and Europe. With additional funding, we will develop imaging biomarkers that can be used in non-ambulatory patients so that boys that are no longer walking can be used in clinical trial. Unfortunately, this is the population that is often left out of clinical trials. Another important focus will be developing biomarkers for patients with Becker's muscular dystrophy, which is another underserved patient population. In these patients, we really want to also understand what the link is between dystrophin levels and ultimately the muscle quality, because at this point, we don't know how much dystrophin is needed to maintain muscle quality. Working with the boys with Duchenne and their family has really been a humbling experience. Our team is always so inspired when the boys come here and we get to interact with the families. Remember, we have worked with many of the boys for many years, and so we really get to know the families. It often feels like a family experience, like we're all in this together to try to make a difference for the children with Duchenne. I'm optimistic that within 5 or 10 years that there will be an effective treatment for boys with Duchenne muscular dystrophy. There are a number of therapeutic targets right now that have the ability to slow down disease progression and improve the quality of life. And our hope is, of course, that our MR biomarkers that are so sensitive that they will help us get to this point even faster.
All right, and now I'm going to pass it over to Dr. Walter for his presentation. As Dr. Walter gets his slides on the screen, this is a reminder that if you have any questions, there is a Q&A button at the bottom of your Zoom screen. And if you click on that, you can submit questions for the end of the presentation.
Eric, thank you very much. It's my pleasure to tell you a little bit more about ImagingDMD, and especially about the biofluid biomarker discovery that we're doing in the biosamples, because we often don't get to speak about that. We primarily focus on the imaging parts, but ImagingDMD covers many different aspects. So I'm going to talk about initially phase 1 and phase 2, and this is data collected over the last 10 years. This is part of an NIH-funded study, and with PPMD has been with us all along the way, as well as many people who've participated in this study are logged onto the webinar, and we'd like to thank you from the very beginning. So what's unique about this project is that we're taking MR data, not only just acquiring images, but we're working with local expertise to understand the muscle biology that's specific to changes occurring in a number of different dystrophinopathies, bringing in data analysis and statisticians to generate models which can be used in clinical trials and develop clinical trial infrastructure. Right now, we support a number of different clinical trials in the United States, Canada, as well as Europe. We're likely to have an impact on clinical trials for a number of different dystrophinopathies, including BMD and non-ambulant DMD patients, as well as trials that are targeting the dystrophin rescue in DMD itself. So one thing I'm really proud about is our goal is to be inclusive. And the goal is really to be able to cover the imaging biomarkers as well as biofluids in participants over all ages and ranges of mobility. So first, let's just talk about DMD, and this has been a focus of phase 2, and now our phase 3 part of the ImagingDMD, which was just recently funded for NIH for the next 5 years. So the goal is really to expand the range of dystrophinopathy patients. Thinking about in DMD that 2/3 of the patients are currently non-ambulatory. And a lot of the clinical trials still rely on ambulatory endpoints. This results in the exclusion of approximately half of the boys from clinical trials. And our goal is to be able to get every boy into a clinical trial that would like to be. We need to develop sensitive ambulatory as well as non-ambulatory measurements to be able to cover this range of mobilities and facilitate that, to be able to facilitate inclusion in clinical trials. So we need transitional outcome measures, which are sensitive across a wide range of disease states and states of ambulation. Starting last year and again into phase 3, we're expanding into gentlemen with Becker's muscular dystrophy, which is supported by NIH, as well as we're initiating a study with PPMD's support into looking at female affected carriers, young female affected carriers. So again, the goal here is to be inclusive and comprehensive. So the goal is to have comprehensive evaluations from muscle structure, function to metabolism. I'll go through each one of those. This is where we're moving in phase 3. This is an example of a coronal image from a boy with Duchenne muscular dystrophy of 10 years of age. This is the coronal image. This is an example of a more advanced boy with Duchenne muscular dystrophy, which has a sagittal image, which has extreme contractures around the hip and the knee, which does not permit this boy to be able to extend his legs out straight. Now, in the previous phases and in lots of MR studies, we would have to exclude this boy due to the extreme contractures. But now using whole body technology, we're able to include the boys who've now transitioned to not only to non-ambulatory, but have such severe contractures that they would normally not be able to extend their limbs into a normal MRI coil. And why is this important? Not only do we get whole body coverage, but we get to be able to image their muscles from the transition from ambulatory to non-ambulatory. These green images, what's shown as green is fat, and all the red is either the remaining muscle or blood. Again, what's very important, we know in young boys, the first muscles which are going to be replaced by non-contractile tissues are in the hamstrings, followed by the quadriceps. But early on, when they start to transition, we start to see the biceps brachia and the posterior forceps will start to have their muscles replaced by fat. So being able to measure multiple muscles across this continuum will help us to develop biomarkers which are sensitive across the entire range of the disease. And this will be true for Duchenne as well as Becker's muscular dystrophy. So just another way to show that, not only are these biomarkers indicative of this replacement of muscle by fat, but can also show therapeutic intervention. So for instance, in this case, this is an example from boys with muscular dystrophy's calf. Again, what is red is muscle or water. And this is the soleus muscle. In this case, what we're measuring is the replacement of the soleus muscle by non-contractile tissue or by fat as a function of age. And one of the most prominent findings that we've found in a number of papers, but the most recent one by Bill Rooney, published in Neurology this year, is that the rate at which the muscle is replaced by non-contractile tissue will depend if the boy is on steroids or not. So for instance, the steroid-naive boy will have the soleus muscle replaced by fat, or 50% of it replaced by fat. It will occur approximately 5 years earlier than the boy who's on steroids. So again, just showing the sensitivity MR to look in different muscles and be able to determine the rate of a therapeutic intervention. What we're very excited about this initiative is expanding this to some of our colleagues, our European colleagues, in this case from Leiden University, which are also experts in MR imaging and biomarker development. In the same year, they published similar results of looking again, fastest ladder of the quadriceps muscle replacement by non-contractile tissues as fat, in this case, as a function of age. And again, using a trajectory modeling, was able to take these trajectories of fatty tissue replacement and relate that to the age in which the boys would lose ambulation. And this is a unique opportunity where we can join forces to be able to address, can we look deeper into these trajectories and find out more about what the lack of dystrophin has in terms of the muscle biology. MR also has the advantage of function. In phase 3, the current phase of imaging DMD, we're looking at the expiratory muscles. Again, here, green is fat, red is muscle. This is an example of a image through the chest of an unaffected control in which we see a lot of muscle around the rib cage. The case here is a DMD boy that's ambulatory, and here's a boy who's non-ambulatory, where we see the increase in the amount of fatty tissue deposition and just bulk fat mass around the chest wall, shown here as green, as well as some of the loss in the muscles, certainly in the non-ambulatory case around the ribs themselves. The other advantage of MR is not only do we get structure, but we get function. So we have the opportunity to look at inspiratory muscles, such as the diaphragm, or at least the effective diaphragm in terms of when the lungs are filling with air during a maximal inspiration test. You can see the diaphragm depress the liver as the lungs start to fill. And this gives us a readout of inspiratory function that we can measure in real time, looking at diaphragm descent and chest wall expansion, and relate that to the clinical measurement of the pulmonary function test. In a subset of 50 boys, this is also tacked on with serial measurements or longitudinal measurements of cardiac function, such that we have good coverage of cardiopulmonary function in these boys, as well as composition. So returning to these trajectories, this is really the basis of this initiative, is if we look at the trajectories of all the boys within imaging DMD, one, the trajectory of the replacement of their muscle by fat, or two, based on their distance and their ability to be able to do the 6-minute walk test, is we see the population is characterized by a subset of boys who progress very slowly and remain ambulatory until the 18 or 20 years of age. Whereas there's another subpopulation which progress very quickly. A majority of these boys are steroid naive, but not all of them. So one of the goals of this project is to understand what is the root cause of this variability. So as part of imaging DMD, and you'll hear a few minutes later about what's happening at Leiden, is we've been collecting biosamples along with each one of their each one of the participants' visits. Now this is an optional study, so not at every time point and not every participant gives a biosample. What we find is, and it makes sense, is most boys in this case will have given urine. We collect urine, blood, and DNA from either fibroblast, from saliva, or from blood pellets. Our most robust set of data is from urine. In this case, we have 197 individuals which have been urine throughout the extent of phase 1 and phase 2 of imaging DMD, and 115 of those are longitudinal. And of course, I don't like doing it, but a lot of the boys don't either, is giving blood. Those numbers start to taper off. But we're very well sampled into the urine. So what we've been looking at to this point is a very limited analysis of the proteins within their serum. We found that as similar to what Eric Kaufman and Yatri Palthoff has, the effect of steroid use on MMP3 and MDC3. We've looked at a couple targeted assays. But we're also looking at lipids to look at dyslipidemia, serum metabolites and creatine to look at changes in muscle mass as well as turnover in the muscle. And DNA, can differences in the primary dystrophin mutation or potentially mutations explain the differences in the rate of progression in those fast and slow groups? And this is in collaboration with UCLA, with Stan Nelson and Richard Wang. We're also looking at the urine proteins. We'd be silly not to. That's the easiest biosample for us to collect, and we have plenty of it, which David Hammers at the University of Florida is looking at the Titan fragments, as well as we're again looking at urine metabolites, comparing metabolomics in the serum and in the urine, and at all times trying to relate this back to the MR-determined disease progression. So one thing, and this is a very complicated slide, and the only reason for me to show this is just to show these relationships can get quite complex. So the goal of the project is to collect enough data and phenotype information that we can really dissect some of these relationships. Bottom line is, if we look at the relationship of the replacement of that quadricep muscle of fat, non-contractile tissue, we see a different relationship between the metabolites in the urine and those boys who are on steroids versus those who are not. And so this gives an opportunity not only to look at the differences between fast and slow, but with different steroid regimes as well as therapeutic intervention. So we want to take this to the next, this initiative lets us take it to the next step. So the first part, as I've already mentioned, it's going to consists of an international collaboration with Leiden University to increase the number of well-characterized natural history samples. This includes both functional, MR, as well as blood and urine samples. Then we take this and do a deeper biosample analysis. So part one simply is to take together these two cohorts and increase the sample size of longitudinal data. As well as what I've alluded to earlier was that with the group for Leiden University has a large cohort of patients that they've been following through a number of different universities, which covers a large span of samples, as well as boys who've transitioned to non-ambulation and on different glucocorticoid steroid status. And so again, this will help us for model development and model testing by increasing the sample size. We want to go deeper. So in this case, we're going to use SomaScan using aptamer technology. And this was shown very early on by Jutri Hoffelt and Eric Hoffman's group, is to be able to identify out of thousands of proteins, ones that are specifically associated with change in age in Duchenne muscular dystrophy. In this part of the project, we'll expand this to 3,000 to 5,000 potential proteins in both the serum and the urine. So the goal is to take the samples, do deeper analysis of the protein levels, combine those, and map the correlations between protein expression and the timeline of disease progression. Now, you can imagine this gets quite complicated. This is just an example of the correlations that we're finding in the ImagingDMD data with a very small sample size of proteins, only about 30 or 40 proteins, about 50 or 60 metabolites, and looking at the differences in the muscle T2s. Again, this figure is not for you to tease through each correlation, but just to show the complexity in the data. And we hope to be able to unlock from this complexity processes which are informative in terms of the dystrophy process and hopefully reversal. So an example of that's already been done by the Leiden group. And if we look at the Leiden group's results from the biosamples I mentioned earlier, they did a study and we're looking at 118 targeted proteins using Luminex type technology. And what they looked at was the dependence of the changes in these proteins on age, ambulation state, as well as glucocorticoid steroid. So out of these 118, they found one that was malate dehydrogenase 2, which was associated with age, disease progression, shown here, as well as, excuse me, it had significant association between the time of loss of ambulation after correction for age itself. So the MDH2 became a nice example of a protein biomarker in the blood, which could show disease progression as well as being a predictor of loss of ambulation. When this range of proteins are increased in this study using SomaScan, we expect a number of overlapping proteins to be dramatically increased, and gives us an opportunity to look into more mechanistic changes that occurs in muscle lacking dystrophin. So overall, the project is, again, to combine natural history studies, dive deeper using the SomaLogic or SomaScan, combine that with the phenotype data and the functional data, map out these correlations, and from these correlations, hopefully unlock new insights into the dystrophinopathies, into the process of muscles as they experience the loss of dystrophin itself. Of course, I think this is actually absolutely the most important slide, is the acknowledgement slide. I'd like to thank PPMD for their support from the very beginning. And of course, I would like to thank all the families who have participated. There's been some families that have been with us all the way from the beginning, and it's been a long time. When you come to the Portland site, you may interact with Bill Rooney, who's the director of the imaging facility. Or if you go to the Children's Hospital in Pennsylvania, DJ Wang. If you come here, you might see me, but most likely you'd run into Dr. Vandenborne, the director of ImagingDMD, or Lee Sweeney, the co-director. But to tell you the truth, the feet on the ground, the people you most likely will run into are all the other people in the picture, such as Allison Bernard, Donovan Lott. And I know every year that they look forward to your return as well as we do. And I know the boys look forward to seeing them. And so I want to thank PPMD, the families, and of course the National Institutes of Health for funding these studies through phase 1 to phase 3. And I would finally like to thank the group from Leiden for their feedback so far. And we're looking forward to collaborating with the leaders there, with Pietro Spitali in terms of biomarker development, proteomics, Hermine Kahn in imaging, and Eric Nixon, who runs the Duchenne Center. And I'd like to acknowledge their support also. So, and finally, thank you for your attention.
Thank you very much, Dr. Walter. That was a great presentation. We're, thrilled to see the expansion with ImagingDMD into both the Becker and the young carrier populations, and really excited to be supporting this work, kind of taking a deeper dive here into understanding Duchenne and disease progression. We have a few questions starting to come in, so I will kind of direct those to you and Dr. Vandenborne. So one of the first questions we have in regarding individuals in the study, are they just on steroids, or do you have individuals in the study that are on other therapies, and is that something that you would share and investigate the changes?
Krista, you want to take that question?
Yes. So in our study, we typically accept subjects that are across the board. They might be on steroids, they might be taking vitamins, they may have been enrolled at some point in a clinical trial. So we do keep all of that information as part of our study. If anybody is in an active clinical trial, that would not be shared, but trials that have been finalized, that information will be shared.
As well as document steroid use and regimen.
Great. Now, regarding looking at both the urine and the serum, what level of, do you expect overlap, I should say, or more differences between those two samples?
Yeah, it's quite amazing to think that you could use urine to scan for serum proteins, but it's really, looking at the NMR results, we can clearly see there's lots of proteins within the DMD urine. And just recently, I think just a couple of weeks ago, that Kay Urlander from Ireland published a study looking in dystrophic mice. And looking at that study, they were actually able to identify 1,000 proteins in the mouse urine from both control and 800, I believe, from dystrophic mouse. And what they found were some of what we had known before in urine is that what you can see is even fragments of huge muscle proteins that are typically seen in the serum in the urine, so titans 1, as well as you could see proteins which come from the extracellular matrix. And so they're going to be certainly not the same exact proteins, they may be fragments, but the coverage is actually quite large, to tell you the truth, and it's going to be exciting to see how much overlap there is in the human data.
And so then there's potential there, if there's enough overlap, to rely more heavily on urine as a source for samples, which would, I think, be beneficial for all the boys if they could provide urine rather than getting stuck with a needle.
Right. As well as, like I said, it's a rich source also of metabolites, so that we can get a lot of information from urine. As well complemented with the creatinine, sorry.
Yeah. And then as you're going through, and I think you kind of, again, alluded to this a little bit in one of the images, but as you go through the process of mapping, the proteins that are being identified, you're going to be able to kind of categorize them or with different groups in terms of whether it be inflammation or fibrosis.
Yes, go through, use a lot of the standard tools that are used for proteomics and metabolomics to look for protein networks and correlation between changes in proteins within a network itself will help to identify different inflammatory proteins versus proteins which are more just structural proteins which are leaking out of the muscle itself, and possibly changes in mitochondrial proteins as the Leiden group has presented.
Great. And then, so when you're, after kind of getting some of the results, do you anticipate being able to look at and predict the processes that are driving the muscle replacement and loss of function. And so taking this data, integrating it with the MRI, as well as, and I don't know how much you have in terms of biopsy data, if any, or if you would look to combine that in the future.
Yeah, absolutely. And in this phase 3 in Becker's, one part of the study is to take muscle biopsy data, so that we should be able to really get a comprehensive view of what's happening on the muscle level, the serum level, the urine level, as well as the MR level. We generally speaking, we feel we can track with MR the inflammatory to the replacement and fibrosis that occurs within the muscle itself. And we would expect to see similar processes in the protein changes. But it'll be interesting to see how we untangle all of it. Like I said, it's a complex work, but putting these different pieces together really helps us to solve the puzzle.
And so another question coming in on looking at kind of the difference between fibrosis and fatty replacement. Do you think that you'll be able to see differences in terms of the protein biomarkers that are seen to kind of differentiate or predict, I guess, the muscle level fibrosis and versus fatty replacement?
Yeah, with MR, fibrosis is a little bit still elusive. So we actually hope to be able to rely on the urine and serum biomarkers to tell us about what's going on in terms of collagen levels in either biofluid, or what's going on in terms of the changes in other extracellular matrix proteins. So I think that's another example where these hopefully will go hand in hand and provide insight for both the protein biomarker discovery as well as the MR aspects.
And so this is, as you kind of already stated, it's going to, help. There's going to be some dot connection between potentially imaging and the proteins. And then you also have all of this functional data, both from ImagingDMD as well as Leiden University. And you think that'll also provide some insights to the functional data that we have as well. And I think most of it is, or maybe you can speak a little bit more to the type of functional data that has been collected regarding, ambulation versus non-ambulatory data that's through ImagingDMD.
Maybe I'll pass that one to Krista, give you a chance.
Yeah. So yes, throughout the course of the ImagingDMD study, we have always collected functional endpoints as well. Those are the typical clinical endpoints that are used in clinical trials. So we do the 6-minute walk test to get up from supine, climbing 4 stairs, the 10-meter walk run. In phase 2, we also included the North Star as well as the pole. So at this point, we have a very rich natural history data set also from a functional standpoint. The Leiden group has very similar measurements they've done, and they have that in a lot more patients than they have the MR data on. So we will be using also the functional data to categorize patients that progress faster or slower. And so by doing that, we can actually expand the sample of the size of our samples. Mm-hmm. And of course, the functional data are always really important to provide context to make sure that what you're looking at is clinically meaningful. So I think that this is really such a unique opportunity by taking all of these different data sets, partnering with PPMD and with an international group to really get the most out of the data. And so I think this is going to have, tremendous influence on the community and really set the stage for a lot of discovery over the next, certainly 5 years.
Excellent. There was a question as well. So I know you've said it's a partnership with ImagingDMD and Leiden. We did have a question, I think, just to clarify if there were any companies involved within the initiative or sponsor involvement at this point. And I mean, just to clarify that there at this moment there is no sponsor involvement. It is just between the two kind of academic institutions.
At this point, that is where we're starting. I am really hopeful that this is really a nice example of collaboration and maximizing the information that has been gathered and really maximizing the contributions of all of the families and boys that participate in these natural history studies. So I'm very hopeful that with that, other partners will come to the table as well, so that maybe can even expand further.
And building on not just insights into the kind of the progression of disease, do you anticipate that, at one point down the line that these serum and urine biomarkers as well could be used to either provide insight as to the, effectiveness of a drug intervention, or perhaps to make predictions about someone, whether or not an individual would be more or less receptive to a particular therapy?
Yeah, absolutely. I think the advantage of a lot of the urine serum biomarkers is we also get some insight into metabolism, right? So, one thing it's the work that Elizabeth Nally's been doing is just the use of, intermittent steroid use. We already know that in the case that she's seen in animals, and as well as men with Duchenne muscular dystrophy, and some other studies, is that the intermittent use of glucocorticoid steroids has a very different metabolic profile than it does versus daily use. And so just from that case itself, just looking at metabolism is one example. You could think of other therapies which are targeted towards muscle mass. And you could see either, in the case of increasing muscle mass with or without increase in sarcolemmal integrity, it would result in a very different protein profile. So it's quite exciting to be able to think about these different therapeutics and what kind of profiles would you predict and what do we observe?
That's fantastic. And then, of course, I think as we always kind of end up touching upon, it's early days within the realm of the microdystrophin gene therapy trials, but similar, could these biomarkers help inform us not just how people are responding, but as we have questions about durability over time,
Yeah.
Changes in individuals, to kind of provide some insight into what is happening.
Yeah, I think that'd be very exciting, especially with a number of different constructs being out there. You could see that some may be more metabolically adept or not. Also with since a lot of these interventions are being done in very young boys, the case of separating out muscle growth versus, again, just protecting from muscle damage, pulling out those different signatures, I think would be extremely important. And it sounds like that's really data to look forward to.
Yeah, certainly. Actually, and I think this question might be more about some of the ImagingDMD work, but are you planning on adding reachable workspace within the non-ambulatory patients? Krista, I don't know if that's something you can address.
We have not, it's always challenging, right? There's only that many measurements that we can do. And so we are right now, we just finished our investigators meeting to try to maximize and really look at the workflow and which data to collect. So at this point, we don't have reachable workspace as part of it. That doesn't mean that it couldn't be added on at some point. So this will really be the good time if people are interested in adding in elements or would like to make recommendations. We are really at the stage that we are trying to finalize what the next stage will look like.
Well, great. Yeah, that is a challenge always whenever we have these studies. The time and the resources can be hard to spread out everywhere. Okay, let's see. And I mean, so another question regarding potential, partnership down the line is: are there current gaps that you think that an industry partner might be able to help fill as you move forward with this project? Or is it just, more about kind of having additional access to samples, whether that's from, placebo groups that aren't on investigational products that would kind of, make that a little more challenging?
That's a big question, many different angles. I do think that having access to additional samples will be really important from my perspective. I think industry always takes a different approach. And so I think there's a lot to be learned from interfacing with different stakeholders in industry. So I think there's many potential contributions that could be made, but samples, of course, are going to be really important.
And I would echo that, especially in terms of it's being more and more difficult to find boys who are not on steroids. So the placebo arm in those trials are looking at, alternative steroids or other anti-inflammatories could be very valuable to fill that hole.
Yeah, I agree with that. That is a really challenging one.
Yeah, especially, I mean, too, with even in the starting steroids early in their clinical care as well. So it does kind of reduce that population.
Yes.
Well, I think that is the end of the questions that have come in from our audience at the moment. So I would like to thank you both for participating today. I think Pat's gonna say some final words before we kind of close down the webinar for this afternoon.
Yeah, Krista and Glenn, thank you so much. We think that so from PPMD's point of view, this is something we've always been interested in. I have to say in 2014, when Elias Zerhouni was the director of the National Institutes of Health, I remember having a conversation with him saying that you have to dig deeper and you have to dig deeper and keep digging until you can really understand the differences between fast and slow progressors, until you can understand who responds to what particular intervention. And so really capturing this is important. I think our goal from PPMD, this is our holiday campaign to really initiate this and get some of this wonderful data from both Leiden and ImagingDMD. I think our hope and our goal is to expand this collaboration. We really want partners in this. It takes a village to do this and do this well. We're really grateful for the support we have for this project going forward, and our relationship with SomaLogics has been for greater than 20 years. I think I've known Larry Gold, who started the SomaLogics aptamer-based technology, to look deeper. And so we're very thankful to have this relationship and really anxious to have partners, so that the more understanding and knowledge we can have and the more the biostatisticians can look at, the more we can shed light on what we know, what we need to know, the gaps that exist, and how people respond. And so this is all about accelerating therapy development and getting to precision medicine, which is, I think, where we all want to go. So I can't say enough that we have always been thrilled for what Glenn and Krista do in terms of ImagingDMD for the project they started, and Lee Sweeney as well. We thought that was brilliant and have seen you grow and gotten NIH support, and we're really thrilled about the NIH grant this year as well. In the middle of a pandemic and all of the issues, we're thrilled to see that support come through. As a community, we wish you the best, and certainly from PPMD's point of view, we thank the foundations who've joined us in this effort. We welcome more foundations to join us, and we think that this should grow and grow and grow so that we do things more quickly and better for Duchenne and Becker muscular dystrophy and the female carriers. I just wanted to ask one question from Glenn and Krista before we close, and that is in your grant that extends to Becker. Is there a limit to the number of patients you can include in the study, or is that just an open door for all Becker patients of all ages and stages?
So we will be looking, the way the grant is currently designed is we will be looking at adults with Becker's. I think we have set ages 18 to 62, just to avoid the impact of aging on muscle as well. And it will be ambulatory and non-ambulatory.
And then you do genetic testing on those. And the reason I asked the question, so you have them, as we look at microdystrophins and some of the expressed in-frame proteins from antisense, so that you might be able to get a better picture of those individuals who are working on in-frame proteins that may be much smaller or sort of resemble some of the antisense approaches that we've got. So I think that this will shed light on what we know about those smaller dystrophins that are in-frame, and hopefully support as the gene therapy studies go forward. So again, thank you, Glenn, Krista, thank you, Lee. I know he's behind this. You have an amazing group with Donovan and Claudia and Allison, and all that group has been so welcoming to the family. So thank you so very much. For all of you listening, thank you for being with us on the holidays. We wish you the best in the holidays. Stay safe, and one day soon we'll be again in person. Thank you. Thank you very much for joining us.
Thank you so much.